So welcome everybody to this webinar, and thank you very much for taking the time to come and listen to us. Today we will be talking to you about the challenges faced by industry around in vitro safety testing, specifically for oncology drugs. We will also present some opportunities and solutions to these challenges, taking you beyond binding and looking into functional iPSC-based safety screening options. We will start with a brief introduction to Encardia as a company, followed by an industry perspective on the current challenges and opportunities in safety testing for drug development. Once we have a clear understanding of these challenges, we will move forward into two iPSC-based solutions, which we feel address some of these challenges. We will first look into a novel multi-tissue functional safety platform, followed by an in-depth look at a suite of cardiac toxicity assays, finishing with some closing remarks. So first, Encardia. Encardia has been around since 2009, starting as an iPSC-based cell-based company, expanding into discovery services later on. We have over 15 years of experience and are a team of roughly 30 people. Our headquarters and labs are located in Leiden, in the Netherlands. We have business development across Europe and America, both East and West Coast. Our business is not limited to these locations, though. We have clients across Asia and the rest of the world. We offer high-quality cell supply, perform customer iPSC services, and discovery services. For today's talk, though, I want to focus on the safety services that Encardia offers. So Encardia as a company is an iPSC-based company. We produce and work largely with iPSC-based cells and are strong believers in the physiologically relevant human modeling that iPSC-derived cells can offer. We want to utilize human biology to support your in vitro safety testing. As mentioned, we offer both the discovery and safety services, but for today's benefit, we will focus on preclinical safety testing. We believe that iPSCs can help you move forward with confidence and streamline candidate selection by using relevant iPSC-derived cell models. We aim to assess your safety of the candidates earlier to reduce late-stage concerns. The models can enhance translation, and of course, we understand that each drug development program is different and would need to be tailored to your specific needs. I will now hand over to Annie, who is joining us from UCB.
So, good afternoon or good morning, everyone. I'm very glad to have the opportunity to speak in this webinar, so I'd like to warmly thank Encardia for inviting me. So, unfortunately, cancer medicine is a growing market, as we expect that it will reach more than $400 billion by 2028. And as for any other therapeutic area, there is an increasing number of novel modalities such as gene therapy or cell therapy and antibody-drug conjugates, which combine the advantage of small molecules with those of an antibody. And so far, there are 14 ADC drugs that are approved on the market, and they are all for oncology indication. So we know that in drug development, the attrition rate is huge, and it's even worse for oncology. If we take a drug that enters into phase 1 clinical trial, it has only 5% chance to reach the market versus around 9% for non-oncology drugs. And if we compare modalities together, we see that ADCs are just between small molecules and monoclonal antibody. So in other words, if you work in the oncology field, it's probably better to develop an ADC than a small molecule because it has a higher likelihood of success. But why do we fail so much? There are plenty of reasons, but safety remains an important reason, and it has not really improved over time. And among safety, cardiovascular toxicity remains an important part, as it represents 40% of drug withdrawal during the first decade of this century, and it can be either functional or structural cardiotoxicity. Now, the cardiovascular system comprises three elements: the heart itself, the blood vessels, and the blood. So a drug can affect any of these three components, and I have listed on this slide a few examples of oncology drugs that can trigger such side effects. And we can see that some drugs, such as doxorubicin, can even trigger several of those cardiovascular side effects. So it's an important problem. Now let's look more particularly into ADCs. And if we take the adverse event reports from the FDA FARS database, we see that among the 12 most common adverse events, 3 are cardiovascular. These are hypotension, cardiac failure, and tachycardia. And if we take together the 3 components, so the cardiac, the vascular, and the blood disorder, it represents 30% of all adverse event reports for ADC. And there is a big diversity of potential adverse events. The most important one is hemorrhage, followed by hypertension or hypotension. And of course, developing a drug with associated cardiovascular effects is not something that we want, because it has a lot of potential bad consequences, can lead to drug withdrawal or candidate drug discontinuation or prescribing restrictions or label warning, and at the end, a bad public perception. So that's not something that we want. But how can we avoid this? It's important to have in place a strategy for de-risking, but let's first look at what the regulatory framework tells us. Of course, in an ADC, you have 3 components. You have the antibody, the small molecule, which is called warhead, and a linker between the 2. So far, there is no specific non-clinical safety guidance for ADCs. There is only a clinical guidance from the FDA, which mentions that we should do a QTc assessment on the payload that is similar to what we do for a small molecule, and they refer to the ICH E14S7B guidance. But I told you before that all ADCs so far are approved in oncology, and for oncology, this is the ICH S9, which is applicable. And this guidance tells that a QT assessment should only be done when there is a specific risk. So I did the exercise to look at the submission package of the 14 ADCs approved so far, and I saw that a HERC assay was conducted on the payload for almost all ADCs except 3, and when it was done, it was always negative. The QT assessment in vivo was done either through standalone safety pharmacology study or directly incorporated into the general toxicity studies. And there was only one ADC for which there was some QT prolongation. And finally, the ECG monitoring was conducted directly in patients, as it is usually the case for oncology drugs. And there were 3 cases for which a signal of QT prolongation was identified. But as I mentioned before, QT is only a small piece of all the potential cardiovascular side effects. So that's why it's important to have a de-risking strategy in place. And first of all, we need to understand the mechanism behind this cardiovascular toxicity from a molecular, cellular, and tissular level, and develop some adverse outcome pathways, because once we understand the mechanism, then it's more easy to establish assays able to capture those hazards. And there are 12 key characteristics that were identified for cardiovascular dysfunction. You can see them on the left-hand side in these pink boxes. And it goes from impairment of cardiac excitability, contractility, oxidative stress, inflammation, etc. Some of those characteristics and toxicities can be acute, but others are chronic. And so typically we have in the pharma industry some cardiovascular de-risking strategy. And this is an example for small molecules, but that can be adapted for other modalities such as ADCs or protein degraders, for example. And it's a mix of in silico, in vitro, and in vivo assays. It comprises ion channel patch clamp assays, in silico modeling on virtual cardiomyocytes, and also human iPSC cardiomyocyte, which is a very interesting assay because it combines different endpoints, electrophysiology, contractility, and we can even include also some structural biomarkers such as troponins or microRNA. But I won't speak about it because it will be the topic by Silke. And all those in vitro and in silico assays are applied to drugs before in vivo assays. So we select the best molecules to push in vivo. It's a way also to reduce animal use. All those technologies have evolved over time. In vivo methods have evolved from snapshot ECGs on restrained animals to telemetry in group host animals, and maybe one day in the future we may be able to completely skip animal use. In vitro techniques have evolved from animal-based models where it was still necessary to kill the animals to take the tissue, such as Purkinje fiber or isolated heart. Now we have moved to human-based models, such as the cardiomyocyte assay, 2D models, 3D models, and now more complex models, such as organ on chips, and maybe one day the whole body on a chip. And I even don't speak about the increasing role of artificial intelligence. And in parallel, analytics have also evolved from ELISA, simple assay, to multiplexed assay combining different readouts. And in the clinical settings, we have now wearables to the hospital at home. So to conclude, for sure, there is an increasing societal and regulatory pressure to phase out animal use, in particular for safety testing. Many efforts are ongoing to develop and validate NAMs, which can be defined as novel approach methodologies, but also some say non-animal methods. But the journey is still long. However, it's only possible through a strong collaboration between service providers, such as Encardia, pharma companies, regulators, and academia, to accelerate the process, but most importantly, improve our confidence in all those NAMs. But all those challenges are applicable not only for oncology, but for all therapeutic areas. And it's even more complex nowadays because of this increasing diversity of therapeutic modalities. And it's important to share knowledge, share good practices through a webinar like this one. And I will stop there. I'd like to thank my colleagues Jean-Pierre Valentin and Emma Pauluc, as well as Encardia team again for inviting me. Thank you, and I hand it over now to Sanne and Silke.
So thank you very much, Annie. I'll now take over and cover some, I want to take the next few minutes to cover a novel multi-tissue platform that we developed to look at functional safety screening of ADCs, but also other compounds. So as we heard, 25 to 30% of drug development failures in biologics and ADCs is safety. That's the biggest cause of failure next to CMC development. The roadmap that the FDA published in April 2025 specifically focused on antibodies and conjugates and tried to stimulate moving away from in vivo and look more at in vitro and see what alternatives we can use to really do the safety testing on human relevant models. But how do we do this? As we've seen over the years, a lot of 2D models have evolved, 3D models, we have organ on a chip, and a question that we very often get from people and companies is, but what model do we need to run? What is the way forward? What is the assay that the FDA or regulators are going to accept? And that is a really difficult question. The FDA roadmap for biologics, it's still new. Regulations keep changing, so it's hard. We still need to identify how really we're going to answer that question and what models we want to use and are going to be the base standard one day. So what we suggested in our discussions is actually let's not reinvent the wheel, but use some lessons learned from other therapeutic areas that already face these same challenges. So we are thinking then specifically about cell therapies, and within the cell therapy group, even more so the TCR-T cell approvals. TCR-T cells with the HLA dependency are really hard to model in vivo. This was also recognized by regulators a couple of years ago when new regulations came out, that indeed HLA transgenic mice offer a lot of challenges and a lot of limitations. So TCR-T cells is actually the first drug modality that I know of that managed to successfully submit IND approvals that didn't include in vivo safety testing. So they did everything in vitro. Looking at those assays, we wanted to see how we could improve them and where we can actually use those assays to also apply to antibodies, biologics, and other modalities that now also want to move away from animal models. So we developed a functional iPSC-derived multi-tissue screen to really support that safety assessment in vitro based on the lessons learned. So just to align everyone, when we're saying safety, it's a very broad term that means different things to different people. But here we're very much looking, or at least I'm talking about in vitro safety, where ideally with an oncology drug, you want a reaction against your cancer cells. So you have target engagement, your drug, whichever this is, in this case, an antibody with an ADC, attaches and it kills your tumor cells. But equally, we also know that target antigens for oncology drugs aren't solely expressed on tumor cells. The biggest challenge is that we know that we have low-level expression on some of the healthy tissues, which is often where the in vivo safety testing comes in. What we now suggest is that we have an in vitro safety assay where we actually look for that reactivity against healthy human cells representative of multiple tissues. So ideally then you don't want no reactivity, as in the middle picture is depicted, but it could be that for whatever reason, if it's either off-target binding or on-target off-tumor binding, you see some reactivity of your drug against healthy human iPSC-derived cells. That means that you then need to dive into further what is going on, if this can be managed by dosing or a further in-depth study to see how big this risk actually is, but it really gives you clear guidance on where the risk is for your therapy. What we have done is we've selected 7 tissues that we feel represent the major organs in the body, as well as tissues that we know are often included in oncology screens or have expression of known oncology targets. So we've included cardiomyocytes, so representing the heart. We have vesicular endothelial cells. We have hepatocytes for the liver. We have the alveolar epithelial cells, so your lungs. We figured that spinal motor neurons representing the CNS was a really good one to include, intestinal cells, and then we went for melanocytes in the skin, mainly because a lot of cancer target antigens are expressed there. What we suggest is then, first of all, do an in silico screen, see what tissues are indeed at risk for your, or could have low-level expression of your target antigen, validate that target expression in the cells and in this panel that we're suggesting to run, and potentially even adapt the panel to include other tissues that you know specifically for your drug are a risk that we want to investigate. And then we do an impedance-based assay to look at cytotoxicity. We do this with the Excelligence, and on the next slide I'll cover briefly the impedance readout, just to align again on what that really means and what measurement that gives us. And then you've got a few optional methods to expand. So you could look at any molecular readouts or biomarkers, and you could even add more complex toxicity modeling, but I would always suggest to do that afterwards and use this as initial screen to look for tox. And then all the data gets written up into an IND-enabling report, ensuring that you can use this data to then submit or use it in your IND submission. So just to briefly cover the principles of impedance, these are plates which have electrodes at the bottom, and you seat the cells on top of these electrodes. An alternating current is then applied, and an impedance measurement is taken. What that means is that you can really sensitively over time measure how the cells are looking. So you'll see a different reading in impedance based on cell number that's there, and very clear morphology. If cells flatten out, the impedance rating will go up, and if they start to round up and die off, then the impedance measurement will become lower. So you have a very sensitive platform that reads over time changes in your cells. It's also dynamic readout. So with cardiomyocytes, for example, you can even measure to capture the contraction and relaxation of the cells at a millisecond speed. So in order to develop our platform, to test our platform, we did a case study. We ordered an EGFR antibody, so cetuximab, and we ordered it as an ADC, I should say, and it had the MMAE attached to it, which is a very potent anti-tumor agent. We then did a bit of an in silico analysis to see indeed what tissues were at risk. And what we did see was that the tissues in our panel, several of those did indeed come up. So with skin, liver and lung being ones that showed at least some background expression of EGFR on these tissues. We then went to confirm the expression. So we used the cells, we did high content imaging. And for example, in the heart, the cardiomyocytes, we saw that they came back negative for EGFR. But for the ones that were reported also in the silico analysis, you saw expression in the lung and liver, medium expression, with some low-level expression in CNS and intestine, and very high expression in the melanocytes. We then seeded all these cells on impedance plates, which is relevant is that these cells all have different pre-culture times to get them to the most physiological relevant state. So we tailored it in that way that at the time of treatment, they were all in their optimal state. And then we did exposure to the ADC for 72 hours in a 6-point CRC with all the relevant controls in terms of control IgG and doxorubicin at high doses for toxicity testing. What you then get out as data is the top left corner is our lung alveolar epithelial cells. And there we see clear ADC dose dependent response. So the top line in dark purple, which is a beautiful curve, actually is the doxorubicin. So that's our killing curve. And you see within about 25 hours, you have 100% killing of your cells. The lines underneath that reach about 50% at 72 hours is the highest dose of the ADC, with the lower lines showing later start of killing. And they're the CRC, the lower doses of the same curve. The bottom graph is the vascular endothelial cells, where we see no response at all of the ADC. So again, we have nice killing of the doxorubicin, even slightly more potent at about 15 hours, but nothing else seems to happen. The cells seem very happy and healthy, which is in line with the expression, because there's no EGFR expression on the endothelial cells. In addition, what you can do is at any given time point, you can actually look at a dose response curve, showing in the right-hand graph at 72 hours, you see a beautiful dose response curve of the cetuximab with no response at all on the control IgG. If we then do that and look at the whole safety profile for all the tissues, we looked at 72 hours and the highest concentration, which was 100 microgram per ml. And most people will recognize that that's quite a high dose and not really clinically relevant. But we can then see that actually for skin, liver, and lung, we see clear various levels of toxicity at this exposure in these cell types, with also a bit of response in the skin for the negative control, but everything else showed nicely, beautifully negative for the negative control. What we did also see, and I'm not showing here, is that when you go down to 1 microgram per ml, which is much closer to a clinically relevant dose and is even on the high end, all that tox disappears. So we overdose the system in order to show that this works, and we can pick up tox. But when we go into lower doses, we can indeed say that this would be a safe molecule potentially for on-target binding. So then overall, I've shown you a multifunctional system that personally I've seen used successfully for TCR T-cell applications as well, due to the challenges there in vivo models. We've picked 7 relevant human tissues in iPSC format. So what you often see is that a lot of these in vitro testing is still on primary cells, but that increases donor variability and makes very long lead times for sourcing cells often. So with this iPSC-derived platform, we avoid those delays. We offer this as a kind of standardized base offering, but the option is there to customize. So if you know that your drug has a different tissue type that should be considered and should be included, that's easy enough added. By having the impedance readout, which is really valuable, we can get a lot of data on a lot of cell types, offering then the chance to look into more detail if any of these should give a response that warrants further investigation. Quite rapid turnaround of this assay because it's fully standardized and it's up and running, so you can include it at any stage of your drug development. And we feel it's a really good alternative in your drug development, specifically for biologics, which have poor translatability to in vivo safety models. I will now hand over to my colleague Silke, who will actually talk to some of the cardiac toxicity testing capabilities for more in-depth on a specific tissue and the risks that are associated with this.
Thank you, Sanne. I would like to introduce now our cardiac toxicity testing capabilities at nCardia. As Annie has stated during her introduction, cardiovascular toxicities can be divided into functional toxicity, like arrhythmia or changes in chronotropy or inotropy, and structural toxicity, which can be some standard cytotoxic effects like apoptosis or necrosis, but also tissue-specific effects like fibrosis, cardiac hypertrophy, or cardiomyopathy. The image on the left from a publication by Baik et al. In 2021 highlights some of the proposed mechanisms of cardiotoxicity associated with various immunotherapies. As Sanne was referring to during her part, they can be either off-target of tumor, which in this case is just like an antibody or a T-cell receptor recognizing some amino acids in a different epitope that is located on your cardiomyocyte. They can be also on target of tumor, so that just like the epitope is also expressed in a different cell type. But they can also be on target and on tumor. In this case, for instance, here in the example, the therapy is start killing the tumor cells, which then in turn release pro-inflammatory cytokines, which will trigger some unwanted side effects in the heart. Besides immunotherapies, any other group of cancer therapies can also induce cardiovascular toxicities, which was very nicely summarized in a review by Jörg Herrmann et al. In 2021. And as you can easily see here from this little table that I grabbed from this publication, really in all different classes of oncology therapies, you find numerous examples of cardiotoxicity. To detect both functional and structural cardiotoxicity, Encardia has developed a suite of assays based on our iPSC cardiomyocyte models that can be used for acute and chronic treatment regimen. The latter is very important since structural cardiotoxicity usually only occurs after prolonged exposure of the drug. Over the following slides, I will show you data from the gold standard model compound doxorubicin, which is an anthracycline oncology drug, which is notoriously known for its cardiotoxicity. Before I do so, let me first introduce our iPSC cardiomyocyte products to you. Our standard cell type for cardiotoxicity assays is our ventricular end-side cardiomyocytes, which are highly pure, have preserved sarcomere organization, and they can be produced at scale in our bioreactors. They show a ventricular-like extant potential and respond to ion channel modulators or cardiotoxic compounds. Their atrial counterpart shows similar purity and express also some atrial-specific transcription factors. They have atrial-like electrophysiology, and they respond to some drugs that are specifically acting on atrium-specific ion channels. These cells can be run in various different assays to detect both structural cardiotoxicity, like cell loss, adhesion, or apoptosis, changes in the gene expression or in the protein expression that are indicative, for instance, of a change of a cardiomyocyte into a hypertrophic stage. But we can also detect functional cardiotoxicity with these cells, and that can be, for instance, electrophysiology, that would be indicative of things like arrhythmia, but then also changes in the contractility and also in the metabolism of the cells. Some of these assays can be multiplexed, and they, especially if they are non-invasive, like for instance the impedance assay, they allow for both acute and chronic exposure and chronic studies in the same plate. As mentioned, one of the gold standard model compounds for cardiotoxicity of oncology drugs is the anthracycline doxorubicin. We therefore often integrate it as a control compound into our various assays. If you look at the data from the impedance readout, you can very easily see that there is a dose and time-dependent decrease in the cell attachment and viability as measured here by changes in the base impedance. If you look at changes in the beat rate, you can see that this pretty much follows the curve of the overall attachment and viability. However, if you look at changes in the contraction amplitude, which is a surrogate for the force of contraction of the cardiomyocyte, you can very easily see that the onset of the effect is much earlier than for the changes in the beat rate, for instance, and also that there is virtually no difference in the severity of the effect among the different doses that we used on the cells. So that means that we have a very specific action here, which is not explained by cytotoxicity alone. A similar effect can also be measured by our microelectrode array if you look at the field potential. And in this example here, we looked at the firing rate, which is similar to the beat rate that we see here. And you can also see that over time and with increasing doses, you see a decrease in the firing rate in the MEA. Similar effects can also be measured in the cell's metabolism. You can very easily see that, again, with increasing doses, we have a decrease in the oxygen consumption rate in the cardiomyocyte. And at the same time, we do see an increase in the extracellular acidification. However, if you look at the changes here in the bar graphs, you can very easily see that you see an effect on the decrease in the oxygen consumption already at a dose where there is nearly no effect on the total cell number here in this assay. And this is in line with actually the effect of doxorubicin, because mitochondrial injury is one of the major elements of anthracycline-induced cardiotoxicity. Last but not least, the imaging data show an increase in DNA fragmentation, which is actually what the drug should do, but also, again, a decrease in the mitochondria, confirming the above-mentioned findings. To detect these types of toxicity, it is crucial to treat the cardiomyocytes for way longer than the usual 15 to 30 minutes for classic hERG or other ion channel blocker assays. First signs of specific toxicity with doxorubicin can already occur within the first 24 hours. However, there are other types of drugs, for instance, some HDAC inhibitors, where these effects can only be seen after more than 3 days of treatment. Luckily, for most of the functional assays, we have both the cells and the protocols available that allows also for such chronic treatment regimen. In my last slides, I would like to introduce one of our latest developments to you, and that is our non-human primate iPSC-derived cardiomyocyte. You would assume that with the general increase in vitro technologies, any kind of in vivo study would be on the decline, but that is actually not true. Especially for non-human primates, the numbers are rising. And one of the causes is that many of the novel modalities, like biotherapeutics, will just not work at all in other classic in vivo species. Especially the human immune system is in many cases so different from that of our standard in vivo species that the NHP is very often the only animal model that will work in an in vivo testing system. So at Encardia, we have developed an NHP-derived iPSC cardiomyocyte model, which eliminates the ethical dilemma around the animal use, and it can be generated at scale in the bioreactors. We use exactly the same process that we also use for our human cells, and we can easily integrate it into standard in vitro test batteries. So as you can easily see here, our NHP cardiomyocytes are also very pure. They have preserved sarcomere organization, and they also function exactly the same in, for instance, an MEA and impedance assay. However, when we were testing our typical batch release compounds like dofetilide or isoprenoline, we could easily see already species-specific differences in the reaction of the cells towards these drugs when compared to the human counterparts. These effects became more obvious when we were looking at chronic cardiotoxicants in an impedance assay, where we were analyzing effects after 24 hours and also after 3 and 5 days of treatment. In this assay, the NHP cardiomyocytes were generally more prone to arrhythmia than human cardiomyocytes, but they were also more resistant to cytotoxic effects of the drugs that we've tested. For the doxorubicin, we observed concentration-dependent cytotoxicity in human cardiomyocytes already after 2 days of treatment, but only after 3 days in the NHP cardiomyocytes. Both species showed, nevertheless, beating arrest around day 2 of the treatment at similar concentrations. So here, definitely the structural toxicity was hitting in after the functional toxicity. In contrast, when we look at sunitinib, a tyrosine kinase inhibitor, and the data here is shown below, we could see a concentration-dependent cytotoxicity after 5 days of treatment in the humans, but no toxicity at all in the NHP cardiomyocytes. What we could also observe was that we do see a decrease in the beat rate, but this is also more pronounced in the human cells. And then what we also observed is that we have a massive increase in the cycle lengths of one contraction in the human cells. We do also see some increase in the cycle lengths in the NHP cardiomyocytes, but this is way less pronounced than in the human counterpart. In contrast, the NHP cardiomyocyte showed arrhythmia after 5 days of dosing that we did not observe with the human cells. The species-specific differences observed in these assays highlight the translational value of the NHP iPSC cardiomyocyte model, especially for the testing of oncology drugs or biologics. With that, I will come to some closing remarks. So what we've learned today is that all classes of oncology therapeutics, from small molecules to ADC and CAR T-cell therapies, can lead to toxicities in various non-target organs. The increasing diversity of modalities makes the assessment of potential toxicities much more complex. Increasing societal and regulatory pressure to phase out in vivo studies, in particular for safety testing, has led to a push of the development and validation of novel approach methodologies, or NAMs. Encardia has developed an iPSC platform to assess toxicity of therapeutic antibodies and ADCs on 7 non-target tissues, revealing the time course of cytotoxicity beyond just binding. We also have developed a suite of assays to detect both structural and functional cardiotoxicity of any modality in either human or NHP iPSC cardiomyocytes. With these in vitro test batteries, safety and toxicity testing of your oncology therapies can be implemented at earlier stage of the development process, decreasing the risk of late-stage failure. Together with our industry, academia, and consortium partners, we aim to accelerate the development of novel iPSC-based in vitro assays. And with that, I would like to thank you for your attention, and we are very happy to take your questions now.